Okay, so we're done um, discussing simple staining, but remember another thing for simple staining. During simple staining, we fix the samples, bacteria, microbes by, of course, you have air drying, and we pass slide through flame to fix. Okay, so we need to heat the specimen. Okay, and then after heating, of course, we flood it with our simple stain. Okay, basic stain. And then after that, we mount it and check under microscope. Okay, so there's another way or another technique of staining. We call it negative staining. So what is negative staining? So take note that mostly inside the cell, inside the cell, it's negative charge. Okay, and we use a negative acidic, sorry, we use acidic stain so we could stain the background, not the cell. That's why the term is negative staining. So most of the time, um, like if we want to stain the cell with chromogen, that is positive staining. But if we want to stain the background to visualize the cell, it is negative staining. So similar with positive staining, negative staining could also determine, of course, the cell morphology, the cell shape, as well as the cellular arrangement in bacteria that are too delicate to withstand heat fixing. So one example, of course, some bacteria, they have a flagella or other structure outside the cell wall, such as capsule. Okay, so those component or those um, parts are actually sensitive to heat fixing. So that's why we try to develop negative staining. Okay. Also, we're determining the accurate size. Um, it's also crucial for negative staining, and because negative staining minimize cell shrinkage. Why? Because we didn't pass the slide through the flame to fix the specimen. Okay. Therefore, again, we minimize cell shrinkage by negative staining. Okay. What else? So negative staining, also again, we use dye. Of course, the dye should be acidic or with negative charge uh, functional group. Okay, and what else do you need to remember? Um, yeah, so that's the idea behind negative staining. So an example of negative stain, we have nigrosin. So nigrosin could um, stain the background and of course give the bacteria. Okay, so therefore we could determine the arrangement as well as the morphology of this bacteria. So this is Bacillus megatherium. Okay, megatherium. So nigrosin, it's dark, and take note, this is the structure of nigrosin, and there are kind of functional group with negative charge. That's why this dye is acidic. Okay. Another example of negative stain, of course, you have Congo red. We could also use Congo red instead of nigrosin. So here we have the same bacteria, um, the Bacillus megatherium. Okay, and similar to nicotine, this also have functional group as you see, negatively charged functional group, making this um, pigment or stain acidic. Okay, so these are some example of negative stains. So we have this bacilli and some spiruli and of course cocci. So it's also obvious, uh, like the shape of the bacteria. It's so obvious to observe using this staining. Okay, so with some modification for um, negative staining, we could create another kind of staining called capsule stain. Okay, so what is capsule stain? So compared to previous staining or simple staining, which only try to stain the bacteria to know whether they are to know the bacterial morphology as well as the arrangement. Capsule staining is a differential staining. So what is a differential staining? When we say differential staining, we are trying to um, differentiate groups of bacteria. So for example, we have capsule forming group versus non-capsule forming group. Okay, that's why capsule stain is a differential stain. So it detects cells that capable of producing, of course, this extracellular polysaccharide known as capsule. And capsule production is important for some microbes because it's a part of their virulence. Okay, so anthracis, like Bacillus anthracis, as just the one that causes anthrax. And of course, we have Streptococcus pneumoniae, the one that is known to cause pneumonia. They have capsule, and this capsule allows them or making them less vulnerable to uh, phagocytosis. So, parang the capsule repels the phagocytosis of macrophages. Okay? So therefore, nahirapan yung macrophages na um, kainin or iphagocyte yung bacteria with capsule. Okay? Okay. So here we have Mave-Val's capsule staining. Actually, this is Mane-Val's capsule staining. And of course, if you look at the protocol, there's no uh, heat fixing. Okay, they just do air dry. Okay, and of course, first there's a grease free slide, and then we add Congo Red. We could use Nigrosine instead of Congo Red, and then after that we um, put the specimen or yung bacteria over the Congo Red and prepare a thin smear. And then we flood the Maneval solution, rinse with water, and air dry and observe under microscope. So under a microscope, this is the appearance of Maneval's capsule staining. So we have here Bacillus sp and Staphylococcus epidermis, like it's a coccus like with capsule, and here. So as you can see, it looked like there is a halo around the cell, so that is the capsule. And capsule, like uh, again, that is made up of polysaccharide, and of course, some also have polypeptide. Okay, and so this is a mucoid polysaccharide. Okay, and another advantages of this technique is it stain, the stain it stain around the cell, so therefore again we limit shrinking, cell shrinking. So therefore, it's better to measure cell size also by negative staining. Okay. So another thing is about this kind of staining. So remember the principle. The capsule is non-ionic, so it's not reactive to the stain. That's why it appears colorless. Okay.